I want to mention the PEI seminar for next semester on February 7th, Boris Aliento from Geo. On March 7th, Rob Nixon, who is jointly appointed in English and in PEI, and the first appointment in PEI is not in science and engineering. They're giving a lecture. On April 4th, Simon Berry from ED will give the seminar. And on May 2nd, Koe from the Woodrow Wilson School will be giving the seminar. Today, we have the pleasure of welcoming
skip it.
No. Professor Howard A. Stone, who is the Ronald and Elizabeth Dixon Professor of Mechanical and Aerospace Engineering. He's also the head of the MAE department. Howard got his bachelor's degree in Davis, his PhD from Caltech, and postdoc at Cambridge, and then did 20 years of service of punishment at Harvard University in what was called then the division. It's now called, what's it called?
The school.
The School of Engineering and Applied Science. It is Harvard version of an engineering school. And the interests of Professor Stone are very wide. He tries to work at the interface of dynamics, engineering, chemistry, physics, and biology. I know at least 10 different people in different departments at Princeton are trying to collaborate with Harvard on those topics. To me, it's quite remarkable. Many things that is done, including work on droplets and red blood cells, since I work at Caltech blood cells. Would you sit down with me to do? No. All right. Let me finish. We got the first enterprise on teaching.
It's not very interesting.
It's very interesting. Okay. It's very interesting. All right. Anyway. You have to know about Harvard is that you've got the big trifecta, which is a member of the National Academy of Science and Engineering and what's called the American Academy of Arts and Sciences. And did that at a very young age. It's unusual. It's one of the most extraordinary professors at Princeton University, Howard Stone.
Okay. All right. So I was asked to give a talk in this series that has the environment. Thank you, Francois, for the invitation. I'm not exactly sure how I'm going to do this, but there are a lot of people to thank. My research group is this wonderful community to me, and they come in all ages. But I'm going to tell you about some work today that involves a number of collaborations at Princeton. One or two problems are collaborations with Bonnie Bassler, Ned Wingren, and Zemmer Gittai. I have a collaboration with Unilever Research, and Patrick Warren is a physicist who works there. And very recently we've done some work with Mike Winton, who's at the Geophysical Fluid Dynamics Laboratory. So I'm going to touch on a number of things, but before I begin, I just want to say what my instructions were so that you understood what I was told when I was asked to speak here. I received an email from Francois Morel and said the seminars are unusual for all speakers or Princeton faculty from a range of departments: sciences, engineering, social sciences, and humanities. There might have been a good luck at the end of the sentence. And he said, just orient the talk towards a topic related to the environment. So I've tried to touch on a number of things that my research group does that have the environment in them. And so how do I understand the environment? First of all, I come from an area of engineering where transport phenomena is an issue. And in transport processes, you often ask about mechanisms. And by that I want to ask, how do things work? Why do things work? How fast do they work? And most of the stories I'm going to tell you are qualitative today, but all of them have a quantitative feature behind them that I've more or less left out. And the systems that we look at involve the environment. Different are the reasons that we can speak about what takes place in water, what takes place in porous media like the soil. And my last example is a climate-related question. And I should say one thing that's a little unusual perhaps about this list is that most people that study the soil are studying small-scale phenomena, and most people that study the climate are studying large-scale phenomena. And the link between the two is not a science issue. The link between the two is a philosophy about what it is to approach a problem. So maybe, and you're allowed to criticize that, by the way. So the rest of the talk is my attempt to suggest what's common about these problems, and I should say collaborations are sought. So I'm hoping that people are going to think of things that we haven't thought about and then want to come talk to us. And now, from my perspective, that would be great. Okay, so I have an outline. What I'm first going to tell you is I was trained in fluid mechanics, so I'm going to tell you about fluid mechanics as a function of length scale. I'm going to try and emphasize length scales. My approach is more one not based on going deep in a very practical question, but trying to identify principles. And so I'm going to show you a few examples of that involve bacteria. I'm not an expert on molecular biology, but the problem we selected is one where molecular biology of microorganisms meets flow. And as part of that, we discovered two things that neither community had appreciated. So I'm going to show you that via pictures. We've studied a problem in physical chemistry that's related to a beautiful problem that allows transport of particles. The technical term is called diffusiophoresis, and I only jest a little when I say this is extremely well known to those who know it well and completely unknown to most of the experts that teach and do research in the subject. And I can say more about that later. And at the very end, I hope in the last few minutes just to say what it means to study a climate problem from my perspective. And we got lucky, I think, to work with Mike Winton from the Geophysical Fluid Dynamics Lab, where we were told about a problem because of Rob Sokolow. Rob's here somewhere. And they mentioned to us a problem that's below the grid scale of climate models, so it's an important issue not in climate models. And I'll tell you a little about it. My postdoc, Bhargav Balabandi, kind of has solved that problem, I think. But it's an example of how we look at problems. Okay, so first I have to tell you, since you're from many different backgrounds, what does it mean to think about fluid mechanics? So first of all, it's really this fun subject. You go outside and you go on the lake and you see a duck. And the duck is swimming and it has a wake. And you see the same wake when you look from high in the air above the earth and you see a ship. And they both have wakes, but they're on very different length scales. The duck is the size of, like this, and the ship is really big. But if you look at the wake, the angle isn't very different. In other words, the angle is almost universal for this problem. So here's a problem where there's similarity among problems that you might think are a little different because they occur on different length scales. On the other hand, you might be interested in swimming, so you can ask swimming questions at the scale of human beings. That's meter scale. You can ask swimming questions at the scale of microorganisms. They're both questions involving swimming. They're both questions involving stirring of the fluid. The physics is somewhat different, but they're both questions about motion of objects through fluids. So you can ask, what do they have in common and what do they have in different? If you're really bored already at this point in the talk, then just if you happen to be out to dinner one of these days and you look at wine tears in a glass of wine, this beautiful periodic phenomena that forms a pattern, that's a fluid mechanical phenomena. The first person who wrote about it was a man named James Thomson. You don't know him. You know his younger brother, William Thomson, better known as Lord Kelvin. But this is a fluid mechanical phenomena which we could talk about. So from my perspective, there are lots of dynamical processes around. And one of the features, if you get obsessed with your own field, so I get to tell you my field's perspective of it, one of the features that's both good and bad is that if you ask about fluid mechanical problems at the scale of the Earth, or fluid mechanical problems at the scale of small devices, here's a small so-called microfluidic device held between two fingers, they're at incredibly different length scales. One is at a scale of 10 to the 6 meters, the other is at a scale of 10 to the minus 6 meters. So they're fluid phenomena that occur over 10 to the 12 in at least length scale, which is a fairly, you might think, large length scale. But the beauty of the problem is they're described by the same differential equation. Okay, now before anyone jumps up and down, if I go to the right reference frame, I'd add a Coriolis term to the equations, but ignore that for the moment. Okay, so one of the nice things, in some sense, but it's deceiving, is that there's a governing principle that describes these problems over an enormous range of scale. That's useful. The equations, of course, are very hard to solve. You generally need computers, but my group doesn't do much of that, but we think about how do you successfully make approximations to approach problems. But it's this universality that is the feature that kind of I like to talk about when I talk about people, and then of course we need feedback from, we all share our specialization, so this is sort of mine. Now, I was a graduate student at Caltech, Francois Morel, Caltech. I was trained in this idea that, fluid mechanics is all governed by the same equation. And I was sitting in the library late one night in the department I worked in. I was a naive graduate student, and I was studying, and then a little while later, very late that night, a cynical older graduate student came in to the room. Now, I'm sure there are no cynical older graduate students, but the cynical older graduate student looked at me and said, "Don't you understand that it's all hopeless?" I was naive, and so I said, "Surely you jest, this is so exciting." And he waved to 180 volumes of a journal, each 300 pages long, each part of a library where there are other journals all in his field of fluid mechanics, and said, "Don't you realize they're all trying to solve the same equation?" And he walked out. Okay. All right. Oh, it was a joke. All right. Now, to appreciate that fluid mechanics both has problems that are challenging and problems that are beautiful, my group knows this picture. I just want to show you this video. The phalarope is a shorebird. It tends to live in shallow water. And when it gets hungry, since it doesn't like to get its head wet, it spins in circles. And of course, if you're a scientist or engineer, you're allowed to ask, why would it possibly do that? What's the role of expending energy to spin in circles? And what the kind of life sciences community or the physical sciences community figured out is by spinning in circles, you create an upflow. And so if you live in shallow water or water that's rich in nutrients, the rotational motion, the vortical motion brings material up, and then all you have to do is peck at the surface of water, and that allows you to eat. Where's the movie's not playing? Well, I'm trying to play the movie. Yeah. So I should say that some years ago I was asked to give a talk at an American Physical Society meeting, and there were on the order of, 800 people in the room, and the speaker, actually I was in the audience, a former graduate student of mine was giving a talk, and he happened to show this movie, and at the end of the talk there were no questions. That always upsets me, so at the end of the talk I stood up and asked a question, and I said in this room filled with 800 people, "Is it true that the phalarope knows more about fluid mechanics than we do?" And he said, without missing a beat, "I can't speak for everyone in the room, but I can assure you the phalarope knows more about fluid mechanics than my PhD advisor." Okay, so my group works on a number of different problems, and I only get to tell you about a few of them today, and they're kind of pictures of the way we try and think about problems, and almost all of them have collaboration, so they're not my group alone. We do some work in subsurface fluid mechanics, problems of flow and porous media. In fact, my graduate student here, Qingyao Lai, is working on a model for fluid-driven fracture, which is a large-scale problem, but we've tried to make a small-scale variant of it. I'll tell you a little about what happens when you have bacteria, which are extremely well studied for lots of reasons from genetics and otherwise, but when you have bacteria and flow. And I'll show you two examples where neither discipline was aware of, one, that there was an interesting question, and two, that something completely unexpected happened. I'm going to tell you about this colloid science problem of salt and water, and how a principle that we're studying allows you to separate particles from water without membranes, using only a small amount of salt. And finally, if there's time at the end, I'll just sketch this idea of what it would mean to study the problem of ice flows in the Arctic using an approximation that gives you insight that climate models can't, because it's below the scale of the climate model. Okay, so that's kind of the story I'm going to try and show today. Okay, so first I get to tell you a little about this problem of microorganisms and flow. The first short story is a collaboration with Zemmer Gittai, but the kind of hero of it was Yi Shen, who was a visiting student with me here, and kind of observed something and then realized it was interesting. So bacteria are micron-sized objects. They often have a single flagella. When the bacteria sticks to the surface, they lose the flagella. But they don't lose the ability necessarily to locomote because they're covered with other appendages, some of which give them motility, most of which give them the ability to stick to surfaces. And the longest of these appendages, which are called type 4 pili, often are located at the poles of a bacteria. The bacteria we're going to work with for the two things I'm going to tell you about is Pseudomonas aeruginosa. It will kill you if you have a compromised immune system. It's about 1 to 2 microns in length and half a micron or so in diameter. Remember that to put the length scale in perspective, for those of you fortunate enough to still have your hair, 100 microns is the width of the human hair. So this is 1/100th really of that scale. I need that visual perspective to help me get through the day. Okay, so here's the question I'm going to ask you in a second. I give you a flow system. I give you a porous system. It doesn't matter what it looks like. Here's an example of a porous system in the lab, a set of channels where the arrows indicate flow. We're going to inoculate a bacterium in one place shown by the red arrow. And then we ask the question, where does the material get transported? Where does it flow to? Okay, I'll tell you what every computer program says that studied this problem. It'll say if you put things up here and all the arrows are the flow, all the bacteria end up in the upper channel. So no one told the bacteria that. So here's the story. Some bacteria have a motility mechanism independent of flagella. In the case of Pseudomonas aeruginosa, that's the so-called type 4 pili, these long appendages that are polar located. And the pili, via a process of polymerization, depolymerization, allow the bacteria to pull itself along the surface. It can't go far. It can't go fast, I should say. And it's generally thought of as a random walk. And so when you ask people why this might be the case, why it might evolve this process, well, it allows you to explore your local environment. Maybe that's a benefit to you. But all bacteria are exposed to flow. It just turns out the interface between molecular biology and fluid mechanics is mostly about swimming bacteria. Most, I think, of the experts would say, well, if the bacteria are on the surface, what's the role for flow? So here's an experiment that we do in these transparent microfluidic devices. The flow is from top to bottom. The bacteria are the little black dots you see. And then the question is, which way do they go? And so in the course of an experiment, some fraction of the bacteria are going which direction? Yeah, so some fraction of the bacteria are walking opposite the flow direction. Okay. They don't go fast, but it's directed, which means they're always going. Okay. And so that was kind of the observation we made that suggested that there's something unusual that happens when you have a mechanical problem linked to a bacterial problem. Now, I'd like to say we discovered this, but we were the second group to notice this. There's a beautiful but obscure paper in the plant biology literature where a group had noticed that a plant pathogen that starts on the leaf ends up inside the plant, and they speculated that it was somehow migrating opposite the flow direction. And we've kind of unraveled, at least for this bacteria and this combination of mechanical processes, how that happens. And it roughly goes like this. The bacteria in solution, if it gets close to the surface, can stick to the surface. The appendages are all over the body and these polar localized pili. But if the bacteria gets stuck to the surface and it sees a shear flow, the shear flow can exert a torque on the bacteria and flip it. And if that were the case, and the bacteria now has the surface motility, this motorized pili, and it pulls on the pili, it's forced to pick some direction, whatever the angle is, but oriented upstream. And some evidence that this is actually what happens is captured by this movie. Alex Persat actually made this suggestion to us. He was a postdoc in molecular biology. The flow is from top to bottom. We put particles in it to see that the flow is from top to bottom. Here's a bacteria. You can see it's about to divide. You can see the septum forming. And partway through the experiment, we change the flow direction. So we make the flow go from bottom to top. And when it does that, the bacteria flips because it's polar localized. So if the bacteria has the motility mechanism, then it's consistent with the picture I just told you. So the question we then asked most recently was, what does this do for bacterial spreading in flow networks, in flow systems? Does it bias the way somehow the bacteria will spread? The reason that's important, right, is it takes one bacteria to kill you, because 1 becomes 2, becomes 4, becomes 8. So understanding how broad they might be is important. So here's our model flow network. We inoculate bacteria where I'm pointing with the laser pointer. We use a wild-type bacteria, that's the black dots. There are some fluorescently labeled, red fluorescently labeled bacteria that have the pili, but they're not motorized, so the bacteria aren't motile. And now when you do this experiment, some fraction of the wild-type bacteria can walk upstream. There's always some probability they break free, and when they do, they end up in the other channel. So there's a critical length scale here, which is what's the probability that you can reach a network far upstream, and that's something we're trying to mathematically model now. But here's an example of a spreading process that's much broader than you would otherwise think because of this interplay between surface motility and flow. Okay? So that's an example I'd call it an environmental example because it had bacteria in it. That's okay, right? I didn't check this in advance with Francois, so he might be. Okay, so here's another one. When bacteria multiply enough, they form biofilms. A typical biofilm would be the stuff that forms on your children's teeth if they forget to brush their teeth. But biofilms are found all over nature. They're aggregates, communities of bacteria, and polymer produced by the bacteria that provide a community structure to the bacteria. So in the pictures I'm going to show you, there's cells and there's polymer. You generally read in the paper, right, that biofilms are bad whenever plants get shut or disease happens, but biofilms are also good inside your mouth. You have many hundreds of species of bacteria that are part of the human health. You have more bacteria inside you than human cells, mammalian cells. So what's the role of biofilms in flow? Is there any place that flow plays a role? And the standard scientists would say, sure, flow provides nutrients. Nutrients let the biofilm grow, and it's clear that biofilms are always on surfaces, and so that's where the growth will happen. Okay, so we do experiments in these small devices. We do experiments at low flow rates, so there's a parameter called the Reynolds number, which is always low. I've colored it in green because the cells you're going to see here are labeled with a green fluorescent protein. With Bonnie Bassler and company, we've done a whole set of biological mutants to understand mechanism. Here I'm just going to tell you kind of the story about how flow affects the biofilm and how biofilms feed back on the flow. When I first made this slide, we did experiments for 10 hours or so. Now we do experiments for about 100, up to 100 hours and the like. Oh, the channel, you see, the channel has little bends in it. To be honest, this was accidental. I don't know how most people feel. Most things in my life are accidental. It turns out that's important. You might worry that it's not a straight channel because, after all, everyone knows channels are straight, but that's kind of nonsense, right? All of you are sitting with your elbows bent. Bends are everywhere, and it shouldn't be our fault that a phenomenon I'm going to show you is associated with bends, and the fluid mechanics community in particular has almost completely ignored that as a geometry to study. Okay, so if the channel was straight, the biofilms are only on surfaces. By that I mean, if this was the inside of a pipe, and the walls of the pipe are the walls of the room and the floor, the biofilm is only on the walls. It's nowhere else. As soon as there's a bend in the channel, then sometime after the experiment starts, you find the biofilm somewhere else. And that's made clear by this movie. I'm going to show you a confocal scan that you take the microscope and you scan from the bottom of the channel to the top of the channel while there's flow taking place in the experiment. We seed the experiment with the biofilm, so there's biofilm on one boundary, actually biofilm on all boundaries, and then sometime after you start the experiment, as you scan through the experiment, you find a string inside the channel, and that string disappears when you go to the opposite wall. Okay, so floating inside this experiment is a mass of biofilm that gets initiated, gets created some number of hours after you start the experiment. Okay. The reason this is important just qualitatively is you go to the literature on biofouling, which is in engineering, it's in physics and in math. And there's lots of mathematical models, and they all have a feature that says flow delivers nutrients allow cells to grow, cells grow on surfaces, and therefore clogging happens by the system clogging from the outside moving in. What this experiment hints at, and I didn't include in the talk, but it's part of it. Our experiments never clog that way. Our experiments always exist for a certain time. Something catastrophic happens inside the pipe, and then the system clogs from the inside out. It's exactly opposite what all the models say, because all the models neglected one fact of nature, which is that biological materials are soft, and soft materials can rearrange in the presence of stresses in ways that we don't completely understand. Anyway, so we were puzzled by this for a long time, so we did experiments in very long channels with sharp corners, round corners, short distance between corners, longer distance between corners. And what happens is that no matter where you look, if you see this so-called streamer-like structure, it's referred to in the literature where it had been seen before as a biofilm streamer. If you see it at one location in the channel and then you look elsewhere in the channel, you see it there too. It's a phenomena associated not with a given location, it's associated just with the fact that they're corners. If you look at all these images, what you start to notice is that the streamer is initiated downstream of most corners, not all corners. It's a corner phenomena, and if it happens at one corner, it's happening at a similar rate at all corners. So a three-dimensional reconstruction would look like this. The biofilm is at the bottom. We cut it off at the top. And then there's a string-like structure inside. And it's very close to the middle. This image kind of hides the fact that there are lots of other filaments of it around. And as I already hinted, though, for whatever reason I left it out of the talk in the interest of time, is that when this system starts to clog, it basically clogs by growing the structure inside. And so that's the mechanism for failure. It's not just limited to Pseudomonas aeruginosa. We've done this with at least 5 different bacteria. They all show this kind of structure. What changes from bacteria to bacteria a little is the time over which it takes for this to develop. And the mechanism behind this is something we don't exactly know, but I think it goes something like this. The material, the biofilm is on a surface. Biological materials are soft. Soft materials have what a mechanics person would refer to as a rheology. If you stress them, they can change shape. And one day we did get lucky that we caught the initiation in this flow of the first element of a streamer. And what you'll see is that you'll form a string-like element, which is mostly polymer. You can see the cells then start to attach to it. And the flow goes along the center line of the pipe. The streamer crosses from one corner to another corner. To model this problem, you have to include a fluid mechanical piece, the flow, and you have to model, or the way we've done it is we've modeled the polymer as an elastic filament inside the flow. So there's a coupling between the elasticity and the mechanics. And everything is driven somehow by some instability near a corner. Frederick. So the model that we've made is a one-dimensional thing. If you look in detail, you'll see there are lots of filaments kind of being generated. But it looks more or less like, on average, one filament. Okay, so we've studied this kind of problem. And the hint that we've and there are a number of groups thinking along these lines, is in order to try and understand some of these problems, you need to think about the biological material in a rheological context, this context of the material itself having its own properties. And there's a fluid mechanical piece that I won't tell you about. Now, is this relevant to the environment? You have to decide. But we've done a variety of experiments for different porous systems to show that this is reasonable for how porous systems behave. We also took a water filter, which is we bought it made of metal wires. And if you seed the system with biofilm, what you see is over time these biofilm streamer-like filaments form between the metal wires. So the development of the biofilm are these string-like elements, and they would then be the mechanism for causing the reduction in flow rate in this environment. So this idea that these structural features exist. We've done it for stents, kind of something that looks like soil, and this example here is a water filter-like example. Okay, so those are two stories. I don't know how I'm doing time-wise. It's mostly stories. Okay, so I'm doing okay. Okay, so the next one is kind of the hero of this is Sang-Woo Shin, who for a number of years worked with Patrick Warren at Unilever and kind of educated us about a problem that has physical chemistry in it, one part of which is well known and one part of which is not well known at all. And more recently, Ora Shart and Soon Shim and a whole bunch of people in my group now are interested in this. So I get to tell you what the problem is, and then what I'm hoping is this might trigger other thoughts you have. So I'm going to show you how ordinary gradients of salt lead to the movement of micron scale objects in a controlled way. So here's Physical Chemistry 101 that you get from all experts. You take a surface, doesn't matter what the surface is, you put it in water. The water has ions in it, say you've dissolved salt in the water. What always happens is most surfaces pick up charge. Glass almost always negatively charged. Surface groups dissociate, so let's say this material picks up a negative charge. The negative charges are really fixed to the solid, so the fluid, which has the counter ions in it, has a distribution of charge, and the extra plus ions are distributed some distance away from the surface because they undergo thermal motion. And so there's a charge cloud associated with the surface. The charge cloud goes by the name the Debye screening layer, or the Debye double layer, or the screening length. The distance over which the charges are distributed net charge is called the screening length. It's normally small, few nanometers, 10 nanometers maybe, very dilute salt system, 100 nanometers, not big at all. Plays a big role in transport. And it doesn't matter what the particle looks like. If you have a polymeric filament, a biological filament, a solid particle, you'll see a charge cloud near it. All right. So if I give you a certain salt, like sodium chloride, which is a 1-to-1 electrolyte, or I give you potassium chloride, which is a 1-to-1 electrolyte, you draw the same picture. The phenomena I'm going to show you in a second distinguishes sodium chloride from potassium chloride. And Bruce, you're a physical chemist, right? Hopefully that sounds a little weird to you. Okay, it does. Okay, so, all right, so here's an experiment from another group. This is an experiment from a group at the time in Lyon, Literic Bouquet, who's now in Paris. Let me describe the experiment to you. They use small channels to show this effect. They're just going to have flow in a small channel. There are going to be two input streams that are the same solution, and then they're going to put particles in the middle stream. Now, if the background solution, the buffer solution, is the same everywhere, then as the colloids go down, if you take a confocal scan across the section, no matter if you look near the beginning of the pipe or later in the pipe, all the colloids are confined to the center, and the boundary is very sharp because Brownian motion can't act to spread anything out at the speed the fluid is going. Okay? So the first image is just to say if it's the same solution everywhere and you have Brownian objects, Brownian motion doesn't play a role in this phenomenon. Okay? Brownian motion of the particles, the little colloidal particles that are, I forget, but let's suppose they're about a micron. On the other hand, if in this problem you put salt on the outside, now it turns out the choice of the salt matters, but I think in this case they use sodium chloride. Then as the particles go downstream, they're slowly spreading out towards the salt. And if you add extra salt on the inside, the particle stream contracts and gets narrower. So you control where the particles are only by adding a small amount of salt. Now, let me just say, is Sang-Woo here, by the way? I forget. I think this experiment was with sodium chloride. If they had done the experiment with potassium chloride, it would have been much, much weaker. Okay? So choice of salt matters. I don't know how many of you are familiar with this. If you say, oh, it's just osmosis, you'd get the word right, and you'd get all the physical ideas sort of wrong, and you wouldn't be able to predict this quantitatively. So one part of this effect, a big part of this effect, is because behind this effect are charged objects, and an important feature of the salt that matters is the the difference of diffusion constants between the cation and the anion. And in sodium chloride, the diffusion constants differ by 50%, but in potassium chloride, the diffusion constants are almost identical. And so the driving force for this, which has a significant part, cation-anion diffusion differences play a role in the transport process. This problem of diffusiophoresis, I think, is lots of places in the environment, in the ocean, elsewhere. A few people are aware of it. I am under the impression that lots of people don't think about it. So that's one reason I wanted to show this. Now, why does it matter? The surface has to be charged. The things in solution, the salt ions in solution, really want to be attracted or repelled from the surface. So there are two contributions to this. One is called an electrical effect, because the difference of diffusion constants give rise to an electric field. Electric field acts on charged particles to make them go. And one is osmotic, just the fact that you have chemical components in the system. If they're at higher concentration in one place than the other, you have a pressure difference which creates a flow. So there's a calculation you can do to predict the speed v of a particle due to diffusiophoresis. And it has in it the fact that everything is driven not by a concentration gradient, but by the gradient of the log of the concentration. Okay, and the way to just show you this idea is you could ask, suppose I give you some porous media, this is going to be a microchannel, and the porous media has a dead end. How do you deliver anything into the dead end, or how do you clean everything from the dead end? It's a dead end, so there's no flow in it. So we're going to do an experiment where the thing we're going to control is the difference of salt concentration between the main channel and the side channel. And we're going to ask, can we put particles into the channel or take particles out of the channel? It's not equilibrium phenomena I want to try and get at. It's out of equilibrium phenomena. The equilibrium problem is extremely well understood. So to give you an idea of what happens, I'm going to show, here's an experiment. The left-hand one is going to be the same concentration of salt between the inside and the outside. The middle one is going to be a factor of 10 difference of salt between inside and outside, and the one on the right is a factor of 100. And if you do this experiment and you're sitting in the first row, you can see white colloidal particles on the right being driven from the main stream, where the flow is from your left to right, down. Do you see that? Sort of. And nothing happens on the left. There's no transport of particles. So, and there's no net flow, by the way. There's no net flow into the side channel because it's a dead end. Okay, so you're using salt to manipulate particles inside a small pore. Why does this matter? The rate at which this happens is not tied to the diffusion constant of the particle. It's tied to the diffusion constant of the ions. So the ion, a 1 micron particle and a small salt ion, their ratio of diffusion constants is 1,000. And so the time it takes when you add salt drops from order hours to order tens of minutes when you create salt gradients. So the rate at which everything happens is controlled by the ions. This one's more easier to visualize. You use two different sized particles. Let's see if I can get this to show up. The green particles are bigger than the red particles, and they move at a faster speed, and they hence penetrate further in the pore. So we've discovered, though we don't completely understand yet, a size dependence to this effect, where you use a salt concentration to move particles. So this effect is referred to as diffusiophoresis. Now, it doesn't matter what the shape of the pore is. You can have lots of different shapes of pores. As soon as you have this salt difference, you drive particle motion. Okay? And you can make it go the other way by just changing the concentration difference. And if you change the salt, you can also manipulate things, as I tried to comment on. Okay, so now why might this be useful in the environment? My group kind of dragged me along, and they created this nice idea, which is that maybe you can do filtering of particles maybe you can remove particles from liquids without filters. And the idea is to use a soluble gas. So this is an example with CO2. When CO2 dissolves in water, it creates two ions. And the ions, because they have different sizes, diffuse at different rates. And in this example, the whatever you want to call it, the hydronium ion diffuses a factor 8 or 9 times faster than the anion. And so when you dissolve carbon dioxide and water and now create a gradient, you now have a means for affecting the particles nearby. Just to give you an idea of how big this effect can be, when you look at the kind of potential differences created when you create a salt gradient with just ordinary sodium chloride, the number you get is 24 millivolts. That's calculation. If you do this with CO2, you get a number almost 4 times as big. So it's a large effect because of this large difference of diffusivities between the small cation and the large anion. Okay, so here's the experiment that shows the idea. On the left is two flow channels. We're going to have just gas in them, and these cross channels have a liquid in them with particles in it. And if the gas is not soluble, or at least doesn't create ions, nothing really happens. The particles stay trapped in the liquid, even though there's gas flow on either side. So it's not interesting. Now, for negatively charged particles, which of course are the standard kind of particles you might buy, now you put CO2 on the top and CO2 on the bottom. And see if I can get this to play. And now what you see is the hydrogen ion goes fast, and because the hydrogen ions are diffusing in very quickly, they're plus charged, they're dragging the negatively charged particles with them, and you move particles away from the interface. So you've manipulated the particles only by contact with gas. And if you reverse this and use positively charged particles, then you can attract the particles to the interface. The particles move away from the source of the hydronium ion. Okay? Everyone see that? Okay, so now how could you use this effect? So then Sang-Woo and Orshart in my group figured out that, well, here might be a way where you might be able to separate particles from water without a filter, and you do it the following way. You make a flow system which has carbon dioxide on one side, diffusing through the walls of a soft channel into a liquid, and then entering an airstream which is free of carbon dioxide on the other side. So you create a permanent gradient of CO2. Wherever you have CO2, the hydronium ion is diffusing, it's trying to move particles away with it. And the experiment that shows this idea is here. And this is kind of where the work stands at this point. So when you run this experiment, you end up with one stream which is particle rich, and the other stream which is largely, though we haven't quantified it, free of particles. And the mechanism is simply contact with a soluble gas. Okay? And in interest of full disclosure, this is a small channel, so the volume of water that you're removing particles from is tiny. Okay, but the principle is a good one, and the question then is, what can you do with it, and can you scale it up, and can you make it a more robust signature? And so we're trying to think about that. But from the standpoint of collaborations, there are lots of environmental issues where there's salt, and there's particles, and there's bacteria, and there are other things. And the question is this principle more widely useful in other areas? And again, just to point out, it's not the equilibrium issue I'm asking about, it's the out of equilibrium problem. Okay, so I guess I'm talking kind of fast. So I have one last example. I have 3 slides left. So 2 years ago, we were visited by Mike Winton from the Geophysical Fluid Dynamics Laboratory. Rob Sokolow set it up for us. And the question was just to talk about climate problems that they thought were interesting. And Mike described a problem involving ice flow. Now, ice flow occurs on scales of hundreds of kilometers. I never work on problems like that. I don't know much about them. I won't pretend any expertise. And the ice flow problem is a big issue, that the ice in the Arctic is seen to retreat, and fresh water flows from the Arctic flow out into the Atlantic, and they're all often areas where the ice has to flow through straits, the largest one being the strait over here, which is very, very large, hundreds of kilometers in width. But there are other places where the straits are smaller and narrower. And it was the narrow that caught the attention of my group, because when things are narrow, which means one scale is large and one scale is small, mathematical approximations often are useful. And in fact, they're exactly the places where you have trouble doing numerics. So the small straits are where the climate models, at least we're told, aren't applied because they're below the grid scale of the climate model. So the question is, if you have this kind of example where you have a strait outlined here in yellow and you have an ice flow from, say, top to bottom, what are the chances that the ice can flow all the way through versus what are the chances that the ice, which you should think about like a granular material of floating ice blocks, actually jams and forms an arch and isn't able to flow through to the Atlantic? And Bhargav Ralabandi, who's working on this, got these satellite snapshots that give you this indication of these chunk-like pieces of ice that are sometimes flowing and sometimes not flowing through this narrow strait. So that's the problem. And the way it maps to the other problems I've told you about is it has a quantitative feature which I like, which is there's a large length scale and there's a small length scale, and they're very, very different. And so in this problem, and this is my next to last slide, we took the literature's view on sea ice, and so in the literature they think of sea ice as floating on the ocean and it's largely driven by the wind. At this length scale, the stresses of the wind drive the ice. There's a much smaller stress exerted by the ocean, and the Coriolis effect at the scale we're looking at is a factor of 10 smaller than the wind stress. The ice has a certain thickness which can change. After all, it snows and things melt. But the strait has this feature that it has a funny shape and it's long and narrow. And it's not the funny shape that matters, it's the fact that it's long and narrow. So we have made a mathematical model of this problem where you have wind stress driving sea ice. The sea ice is held back by the boundaries and it's trying to move because of the wind. And the mathematical model allows us to make a picture of what might be happening when the wind moves, say, from your left to your right. And the Bhargav's beautiful calculation has both an analytical piece and a 2D simulation piece. There are some conditions that lead to this formation of a bridge between the yellow ice field and the blue, which is the open water. And it's that feature that we've been able to make some progress on, including, because the geometry is long and narrow, an analytical approximation for when this might happen. Okay, so those are my short stories. I've tried to tell you a little about kind of how I try and get through every day, which is good and bad. But it's from the standpoint of thinking about dynamics problems and working with others that bring a lot of expertise. And I've told you about a couple examples that I think might have environmental relevance because they have bacteria and biofilms, but they couple mechanics through flow. I told you about this funny problem involving physical chemistry that I think is not very well known to most physical chemists, but I tried to give you examples where it's a very powerful driver of transport processes. And then just very briefly, I realize I probably talked much too fast, I tried to give this idea of how we went about trying to think about an ice flow problem at the scale of many kilometers, because it has a distinguishing feature, which is that it has two distinguished length scales that allowed us to make a simpler mathematical model that we solved numerically and an analytical model that Bhargav worked out and he's still teaching me about. Okay, so with that, I'll answer questions. Yeah, I can, happy to answer questions. You know, if there are no questions, you can always ask me about the duck. The shorebird, it's not a duck, it's shorebird, sorry.
Okay, so you have two examples with the bacteria. There is no interest in the electrostatics or in the streaming potentials on the side, and you still have no role to play in everything you've seen.
Oh, so in the bacterial question you're asking, is there a role for the streaming potential? You're saying the fact that because you have a flow, you create an electric field, and that might act on the bacteria.
Bacteria have surface charge, the pili have surface charge.
So it's a great question. I'm silent because I haven't ever thought about that at the scale of the bacteria for this experiment. There is a small literature on the effect of electric fields, I know, on bacteria to try to understand even can bacteria generate a potential difference between their two poles to somehow do something. I'm not aware of anyone that's looked at that in the kinds of problems we have, and we certainly haven't. My sense is it's weak, and the only reason I say that is the flow speeds we're working are so much faster than the kind of effects that you might get from a streaming potential. I don't think it would play a role, but we'd have to sit down and look at the numbers.
But Brownian motion and the electrostatic forces might be kind of similar. So.
Yeah, but in the bacteria problem I showed you, there's very little role for Brownian motion. Everything is driven by surface motility, which is mechanical, and then the flow is at a speed where Brownian motion plays no role. Frederick?
I have a question about the filament. Like this picture we're looking at, is it a one-dimensional filament in an essentially two-dimensional world?
Well, it's a three-dimensional world. It's so inside a pipe. And so what you have to picture is this thing that we discovered is that you have this material growing on the surface. It's a soft material. In the presence of flow, there's an instability. And I showed you a movie where about halfway through the channel, a filament comes out into the flow and tries to go downstream. And that filament, which is originally some polymeric filament, then has a shape or a position that'll be governed by some interplay between the elasticity of it and the fluid mechanics. And it's floating inside the stream. And then once it's there, it starts to collect everything coming by.
It's a 1D filament or a 2D, like a sheet?
No, it's effectively a 1D filament. That was this. It looks like a string. It's often referred to as a streamer, so it looks like, I think I showed a video maybe. So here's another image of it. So this image shows the filament. It shows it going from corner to corner, and there's a color code on top of it because we did full three-dimensional numerical simulations and some asymptotics to understand the role of the flow that might be giving rise to why things happen in the middle. You're allowed to ask, why the middle? You know, if I talk to people from molecular biology, they will often ask, you know, maybe some of the bacteria know about stresses being different at the corner, but the observation we have, it's generally halfway through the channel that you first tend to see the structure, and we've suggested a fluid mechanical reason for that. Yeah?
It's interesting that the biofilm is going from corner to corner while the streamlines are obviously not
Right.
corner. So
Right.
is any thoughts on why the
Oh yeah, I can tell you that. I have to be careful. I need a string. The only string I have is the microphone. Okay, so so the flow follows the path of the channel. Okay, and then I showed you this movie that's direct evidence that the way this comes about is a filament being formed inside the flow, right? Okay, so if you have a filament in a flow, and it's the flow is along it, the filament is partly under tension, right? So now if the filament has to follow a curved streamline, just hold that end over there. If the filament follows a curved streamline, if you look at the forces along each of the ends, one is going this way and one is going that way, and there's a net force that's pulling the filament across the streamlines. The filament can't, if the filament is under tension, it can't follow the streamlines. It's the interplay of elasticity and fluid mechanics. And it turns out in the mechanics world, there's an old literature going back 50 years that's fluid elastic interactions. It originally created airplanes, right? What's called aeroelastic flutter of wings. In the modern variant of it, there are a lot of small scale problems, that's things my group works on, where there are elastic components and fluid components. And so it's a fluid, what you'd call a fluid structure interaction problem. The filament can't follow streamlines if it's under tension. It's not at equilibrium. Does that help? Yeah. Yeah.
Is there a minimum size to the particle that can be transported?
Did you like it as a phenomenon, by the way? Yeah. So the question is minimum size. We were just talking about this in my group yesterday. The characteristic length scale that sets everything in all these physical chemistry problems is the screening length, the device screening length. And at, millimolar or micromolar salt concentrations, the screening length is, a few to tens of nanometers. The normal approximation scientists make that study this is when the screening length is small compared to the particle size. So anything bigger than that is fine. But at smaller length scales, I think it's going to happen. It's just a question of the rules are going to be a little different because the object is the scale of which the charge is distributed.
So the follow-up is, if you actually say DNA, so you could actually drive DNA to one end.
Yeah, so we've talked about that. We have not done that. But
So it's not a crazy idea.
I don't think so. I mean, certainly all the principles used when people talk about electrophoresis of DNA are these same kind of ideas, except you have an applied electric field. Here, the funny thing about, it's this very funny feature. If I take ordinary water, no electric field, I dissolve salt in it, and I create a gradient of ions. Because if the cation and the anion have different diffusion constants, you get a potential difference. So you get a local electric field in the solution. That in classical terms is, what's the term? The diffusion potential. That's what my group is telling me. It's classical. But that effect can play a big role in these out of equilibrium problems. Yeah, Phil.
You spent a lot of time on the space scale.
Yeah.
What about the time scale, in particular the ice bridges? Do they form fast enough to avoid thinking about seasonal temperature changes?
Oh, yeah. So the question on time scale, well, they must because they form in the environment, right? So
Depends on the width of the landscape.
yeah, so the scales in this problem are set by wind stresses, which vary seasonally, and they're just an input into our model, which has a couple dimensionless parameters, including one that is set by the rheology of the ice. So we haven't tried to, make this wind stress vary with time. We just said there's a typical wind stress, and we pick different values, or you could pick different values for different times in the season. Does that help a little? I mean, yeah. I get nervous and don't like giving talks, so yeah, Francois?
So go back to the question of length, scale,
Yeah.
third
Yeah.
phenomenon. Then if one exists in seawater, where essentially the screening length is very, very short. If you have high ionic strength, the things can happen.
Okay, so yeah, you might think there's no application of that. I'll just say that I mentioned that there was a very beautiful paper from a French group, Litteric Bouquet's group, about 7 years ago. And their paper, which I showed you one figure from, they, and I think like me, I didn't claim anything new in the sense of discovering a new phenomena, but pointed out that there's features of the phenomena that people don't appreciate. And at the very end of their article, they commented that in a variety of problems involving the ocean, a variety of problems involving the environment, you mix fresh water and seawater, and at that boundary you get large gradients of salt. And so in those situations, maybe you should start to think about, is this phenomena playing a role in some of the phenomena you see there? I will say that in the oil field industry, which is a very well-funded industry, as you know, there's a tertiary, or I forget if it's secondary or tertiary oil recovery phenomena, that where you pump salt water into the reservoir to help recover resources. So we have a very recent review article in this area of saline-driven oil, I forget what it's called, oil recovery. And they have a whole list of mechanisms for what might be going on. This is no joke. And they don't mention this one. Right? And this is the one that drives transport, right? And so I think it's just an example that there are phenomena there that the out of equilibrium problem is just not as well understood, I think. And so I think there are probably other places, but I'm no expert. That's why I'm hoping that someone here who is an expert might suggest some problems to us. Oh, that's my...